Tack så mycket. And just a small correction, I'm speaking in English. I hope that will be okay. Biosergen started as a Norwegian company, but in the walk onto the Swedish market, we had to establish a Swedish company as well. So our mother company is Swedish, and we have our office in Solna, close to the Karolinska campus. Okay, we have our disclaimer. Leave that as it is. And I just want to walk into the company as such and talking about our model and our vision. The mission we have for this company is to develop BSG-005, which is our candidate that we are currently putting into phase 1 or have in phase 1 trials in Australia, and which will continue for a couple of months more. On the long term, we intend to be one of the leading companies within this business of fungal infection, particularly the severe ones, which is called invasive fungal infections. Our business model is trying to do it as lean as we can, and we outsource everything that we can. And we will do our continued research on other formulations or other versions of our molecule, and actually with time being a company that has a limited sales force because it's a very slim market. It's very specialized. And what do we really do? We are focusing on life-threatening, difficult to treat, or resistant invasive fungal infections. And we are looking also at the safety of the patients. Clinical development of this product, which is coming from what we call the polyene class of antifungals, is a very interesting new product coming to the market. The main issue is that BSG-005 is fungicidal, and fungicidal means it kills the fungi in question, whereas those who are not in this class are mainly fungistatic. That means they inhibit and keep the growth down of these fungi. And that's something of interest because we know that there is a growing resistance towards antifungal agents. I will get back to that a little bit later. WHO has already mentioned this several years ago and just been out there again. We are also developing other formulations, so we're looking at a long-targeted formulation. That means we will deposit with a special formulation a major part of the dose that we are administering to patients in the lungs. The reason? Lungs is usually one of the first places where an invasive fungal infection gets a place and can establish itself. What we have done is we have done some gene editing of an old product called nystatin in this class of drugs. We made, I think, more than 20 different versions and tested them, manufactured in small scale, tested them in vitro and in cells, of course, and picked out the one that seemed to have the best profile. And that's why BSG-005 is the one that we have chosen. We have done our basic research. It has no genotoxicity. It doesn't have any kidney toxicity. And it's free of cardiovascular, central nervous, and respiratory system adverse effects. The way it works is that it binds to a product in the cell membrane of the fungi, which is called ergosterol. The way it works with that is that it makes pores in the cell wall and allows ions and other stuff to leak out, and that is what's killing the fungi cell. And this is a unique thing for polyenes, that we actually kill the cells. Is it a major issue? Yes, it is. More than about 1.5 million deaths per year are caused by fungal infections. That's something we don't think of. And of course, it's mostly in also underdeveloped countries, etc. But it's still on the global side, something that is higher than most of the other cancer types that we know of. And the only one being more prone than fungal infections, invasive fungal infections and so on, is lung cancer. So it's really a growing area, a field with a high medical need for new products. One of the other things that is about this is that it basically hits us when our immune system is not working properly, which is the case in patients who are treated for cancer. It's in patients who are having transplantations, whether it's organic, solid organic transplantation or hematological transplantations, people with HIV and other diseases such as diabetes, and where you are treated also with steroids. The other major thing is resistant strain development. There has been a lot of use of some of the antifungal drugs in industry and of course in hospital areas. And that is something that has created this possibility of resistant development, mainly for those products who are not fungicidal. And that's what we are seeing growing at the moment as one of the major issues within this. The most important thing at the end of the day is the mortality. And the mortality of invasive fungal infections is high. Depending on the strain we're looking at, depending on the place in the world you are, mortality is anywhere between 20 and up to 80 or 90%, i.e. patients die from this. And just as an example, you know, the invasive fungal infections with Candida, which is a type of fungal strain well known, in hospital in the US was reported to be about 25%. And this is high, even in a country as the US. And how many of these invasive fungal infections do we actually see? And what we're looking at is, if we just take the most prone fungal strains, like candidiasis, it's about 750 patients per year worldwide. Aspergillus, which is a slightly more severe fungal to be infected with, has about 300,000. Cryptococcus is also a bad disease and very often goes to the brain as well. And here we are about 200,000. And there are other types, including the super resistant Candida auris, where there is in total about 500,000 cases a year. So it's not just a small thing. And that's why if you look at the lower part of this slide, you will see that the World Health Organization, it's very often in the news, and this is several years old, but it was just in the news again a few weeks ago in all the major journals and from the WHO again, that this is an increasing health threat. Right? So there's a large medical need for new treatments. And how does it work? How do you get infected? Any normal person like you and I, which are in a reasonably healthy position, we can deal with fungal infections. Or I would like to say fungal spores that, as you can see in number 2 on this slide, that very often comes to your lungs as the first place. If it gets established because your immune system is not working normally, then you have an invasive fungal infection, which is critical for any patient at all. Very often, patients who are with cancer or have been transplanted or for other reasons have a low immune system. They get these infections. And this is typical, the weak immune system that makes this work. So patients that are getting these infections, they get very sick. It goes relatively fast once it has established. It is very important to start to get the diagnosis and start the treatment within the first 4 days. If you miss it, there is a higher risk for a bad outcome. So this is very important to get a fast start of treatment and diagnosis. But what we see is that there are very few treatments available out there which can kill all these fungi. That's what we have been talking about as well. And the main thing here is patients who in this way get infected with these fungal strains, they don't die from their cancer. They don't die after their transplantation or whatever they have. They die from a fungal infection. And the mortality is high. So it's a well-known problem, and it's being spoken out by major organizations. And that's why we are so eager to get on with our product development. Just a little bit on the drug market. There are basically four groups, four classes of drugs. There's the polyenes, there's the azoles, the echinocandins, and then a mixed group of other things. Those that are most important are the first three. The polyenes, to which BSG005 belong, has also amphotericin B, candidiasin, nystatin as main parts of this one. The azoles has some of the well-known fluconazole, ketoconazole, miconazole, and so on. Echinocandins, most well-known is caspofungin, but also other products. But they're all within that same class. And you can see the total market is expected to be around $16 billion. And they're growing, 6.6, 6.8 percent every year. That's impressive, and it's a problem. Just getting back to BSG005, one of the advantages by being a polyene, which is similar to amphotericin B, is that we have a very broad spectrum when you test it on what we call in vitro test systems. That means in fungal cells. I'm sure you've all seen these small agar plates where you put it out and you put your drug on it, and you can see around it where it kills or inhibits the growth. So we have a very broad range of action covering basically all the also difficult to treat ones. When we look at these Aspergillus, Fusarium, Mucor, and Cryptococcus mentioned on this slide, they are difficult to treat fungi strains. When we look at the other groups, azoles and also echinocandin, they have not the same broad spectrum of activity. They do it in some strains, they're good at it, but they also have their problems. And the second thing is, of course, safety. Are there any toxicity with these products? Is there any toxicity with amphotericin B and BSG005? What we also have done is we have tested this in a mouse model where you give them a product called cyclophosphamide, which decreases their immune system. It's like a cancer product. Then you infect them with the Candida strain or the Aspergillus strain, as we have seen here, all working according to a fixed protocol. And then you give them your product for 7 to 9 days, and then you look at these animals for up to 30 days. And the main thing is survival. Who survives? How many survives of these animals? And what we have seen in the left-hand slide in candidiasis, where we are comparing us with equal doses of amphotericin B, ambisome, which is another formulation of amphotericin B, and then our own product at a similar dose, simply for us to see is there a potency difference. And what you can see is that BSG005 is on the top with a 90% survival, and ambisome is down at 30% about survival. Similar on the aspergillosis side, where we also see that we outperform ambisome in this model with a survival of 60% compared to the 20%. So what we're looking at here at equal doses is a potency difference of maybe 3 to 4 times. And that's important. We have also done it with what we call clinical doses, because we don't we have to test it also in that situation in animals. And here we have taken a number of other products in as well. So we have tested voriconazole, and we have tested caspofungin and fluconazole. And then we have done exactly the same. We have given them cyclophosphamide, we have infected them, and we have started treatment with all of these. What's important to note is that we have tested Ambisome at 2 dose levels. 2.7 milligram per kilogram, which is a typical start dose in humans, and also 5.4, which is close to what we normally say is the highest dose of Ambisome. So that's the clinical doses. And then we have given, in our case, 0.35 milligram per kilogram and 0.7 milligram per kilogram. So this is around there we were thinking that a clinical dose would be without knowing it. We haven't been in patients before. And what you can see is that we do almost as good as Ambisome on the highest dose compared to our dose, which is, please look at it, it's 7 to 8 times lower dose. So it fits with what we knew from the previous slide, that we have a potency difference. And similar to the lower dose of Ambisome and our low dose, we see also about 10% difference. But it's a very important note here. And we do as good or better than the alternative medications from the other groups. One of the major issues with amphotericin B and Ambisome has been nephrotoxicity, i.e. toxicity on the kidneys. And we have, of course, tested that because it's a key point. In some studies, 50% of the patients who are started on Ambisome has to be taken off it again for safety reasons. What we have done is we have looked at an enzyme marker in the kidneys, and we have looked at, it's called NAG, and we have looked at different doses of Ambisome, amphotericin B, and BSG-005. And as expected, and as we know from the literature, you see a dose-dependent increase in the damage to the kidneys. Whereas with BSG-005, we reach a certain level, and then there's nothing more. It stays on the same level, no matter the dose. Do we have patents? Yes, we do. We have our original patent, which is actually running out in the US in 2033 expected. And we have, with our orphan indication that we have received in the United States from the FDA, we have a 7-year market exclusivity on top. Similar in the EU, we have the normal 10 years, but orphan would give us another 2 years, and so on in the other countries. And on the patent side, we made, more than a year ago, we applied for a patent on one of our new formulations, which I will mention in a short while here. And we are right now dealing with more patents on the formulation that we have. We are in the clinical phase now. We started our phase 1 trial in Q1 of this year in a standard setup where you do a single ascending dose, and then you do a multiple ascending dose, and you watch safety and tolerability and pharmacokinetics. It's very important to do the pharmacokinetics because it gives you a chance to see what happens over time when you give more doses. So we have a single dose component and a multiple dose component. And the single dose component we have completed now after having run 4 cohorts of 6 subjects, healthy volunteers. It's a double-blind study, so I only have, what should we say, blinded data available, and I will just mention it a little bit during this presentation here. So that's where we are on the phase 1 healthy volunteer study, and our goal is to get the SAD top lines out in December, and then we expect to have our MAD, or multiple dose, subjects out in top-line data in April next year. And then we have started to look into patients, because once we're in patients, we can see whether it works or not. So that's very important. One thing is that it's safe and tolerated and so on, but it's the efficacy in patients that we are looking for. And here we are looking into phase 2, and we are dealing with generally difficult to treat or resistant strains of fungals that we think we have a better chance with compared to many other products. So that's why we're going in with 2, 3, or 4 studies. We haven't decided finally yet in phase 2. And we will deal with strains called Mucor, Aspergillus, Candida auris, Cryptococcus, those that we also have mentioned previously. And the main issue is to document the clinical effect of BSG005 in these resistant or difficult to treat fungal infections. And this is what we are aiming for. We expect that we can have the first patient totally recruited and treated in Q3 next year. We need to have our phase 1 data completed, reported, and so on, and then we're continuing directly into a patient study. So where are we on the financial side? Well, we raised 42 million Swedish in June 22 this year. We will use this capital to finalize our phase 1 clinical trial. We will start the phase 1b/2 patient trial in one of these difficult to treat indications. We will use it to do the pharmaceutical development required when you go from phase 1 to phase 2. As we have also seen from other presenters today, there is a step up in what you have to deliver to patients when you get into patients in phase 2. And we expect that we will have the warrants, which was coming out with the capital raise, and who mature in August next year, 2023. If that all works well, then we will have additional 38 million Swedish, and that added to our cash flow and to cover further phase 2 development until the end of 2024. That's basically where we are on the financial side. So we are doing fine. We have the cash available, and we will of course use it as best possible. Here is our management. We are also lean. We are 3 persons in the management. Myself as the Chief Executive, Niels Larsen as our Chief Financial, and Tine Koller Olesen as the Chief Operation Officer. And with these people together, we have, I don't know how many years, far too many years, I would think, in the industry. But that does cover what we are really needing here. Very little bit on our board. It's a large board for a small company, but we have really decided on getting people in with experience, either into the drug development, into the financial, or into the commercial or environment around research and development. Torsten Gosch is our chairman, and he's with Rosetta Capital, who is one of our main investors. And he has been with the company and supported the company for the last 7-8 years. And the other major investor is Mathias Klintemar from the Østersøstiftelsen, who has also been with us for a long time and is a strong support. Just last slide, I think, on the shareholders. Again, Østersøstiftelsen is the main shareholder. Rosetta Capital is there, and then we have Sintef, we have Karolinska, and we have others. So that's the major owners of shares in our company. And I think that was what I wanted to say, Jan.
Thank you, Peter. Yes, to clarify, the founder is Norwegian.
Yep.
And you are from Denmark.
Yep.
And your headquarters is in Sweden.
Yes.
So it's a Nordic company then?
Definitely Nordic.
Yes. So the drugs that are coming out now in this field, antifungal drugs, why is that? Because nothing has happened the last 30 years. Why now?
Well, because fungal infections, invasive fungal infections, are short term. So they are not like Alzheimer or any other of these kind of diseases, lifelong or very long treatment, etc. It's a short one, and that diminished the interest from big pharma. So they have basically drawn out from everything that had to do with fungal infections. So there was, from those parts, no change, in no new things coming out. So it has really been on smaller companies, biotech companies, to actually work on this. And that's still the case. So therefore, for many years, there were no new products coming out. And that meant you would, for I think it was more than 10 years or 12 years, we didn't see any new product coming out. And now it starts to raise because it is obviously, very obviously, a threat to the general population in many parts of the world. Also in our countries, even though we don't see it as bad as in developing countries, but it's still here. People still die in developed countries from these diseases.
Okay, so what regions are we talking about here that should be afraid of this fungus? WHO has selected some fungus that are dangerous.
Yeah.
And in what areas are the danger the most?
Well, basically, those that are most difficult to beat is some of these that we are actually focusing on. So Aspergillus is always a difficult one to treat. There are often also strains that are resistant to all the drugs, basically, or almost all. And similar, the newest one on the block is Candida auris, which actually was only started to come up in 2009, which is 13 years ago. And started in Japan, and then it spread out over the world, and now it's here, it's in Europe, it's in the US. And the difficulty with Candida auris is it's super resistant. It's resistant to really many drugs in this area. We did some very limited and very initial in vitro testing with BSG-005 towards a Candida auris strain that was resistant to several of the azoles, echinocandins, and ambisome. And what we could see was that we were able to handle it. And the difference between why we can do that, I cannot explain, but I think we have a potency difference. That is the way it works for something like that.
Yes, so phase 1 now for BSG-005. What can you say about this study so far?
I can say that this is a double-blind, placebo-controlled study. So I don't have any unblinded data. They are being collected and being cleaned up right now. And we need to lock the database before we can start working on these data. What I can say is that we have a safety review committee that evaluates every cohort and decides on the safety data and the pharmacokinetic data, and they really decide on what we can. And from these blinded data, the only thing I can say is that we have not had any reports on kidney or liver damage or anything like that. And that was a major issue from us from the start. And we'll just have to wait and see for the multiple dosing that is starting now in early December, in 2 weeks from now, whether we can keep it on that level. That would be, and if that is successful, then we are really far ahead with this one.
Yes, and so what can we say about partners? Have you noticed any increased interest in the company after the WHO's reports and everything that says in media?
Not basically because of the WHO, but we have contact to several companies that are looking at what can BSG005 do in this field. So we are working on several companies, one large scale and two medium scale companies.
Okay, so there's something going on there with partners. We have lunch waiting, and we're all hungry. So thank you, Peter, for coming to us here in Sweden.